having a disease, a disorder, than having a disorder that actually has all this in place already, or unfortunately for the patient. And so I thought that at first what I would do is that I would go back and present some clinical perspective on DBA, although I'm not a clinician, but I've been hearing so much about the disease that I think I can give you some hints about it. So the clinical features of DBA are really the erythroid hypoplasia. That's how it was described at first. When you look at the bone marrow of a patient, you will find that the bone marrow is pretty normal except for the paucity of erythroid precursors. Then there are congenital anomalies, skeletal and growth defects. I won't talk about it today, but the growth defects exist in DBA. It has been discussed and argued for some time because the first line of treatment is steroids. And when you give steroids to patients, of course, there are skeletal and growth defects that appear because you engage into some fractures and some growth defects due to the steroids, but they exist in these patients. And here is a congenital anomaly that you can find in a patient with a triphalangeal thumb or TPT here observed. And as I mentioned already, and I'm going to go back to that towards the end of the talk, there is a cancer predisposition in these patients. So the definition of DBA was described first by Joseph in 1936, and then by Diamond and Blackfan in 1938 as a pure red cell aplasia. That's the classic definition, that is a moderate to severe macrocytic anemia. Macrocytic, the red cells are bigger, and there are red cells that are bigger. They are not the reticulocytes, because these patients have reticulocytopenia. Usually the reticulocyte is 20% bigger than the red cell, but here the macrocytic is due to the red cell. As I mentioned already, these patients have a normal cellular bone marrow with a paucity of red cell precursors, and usually it presents at birth or at maximum less than a year. So there is, I didn't see that, sorry about it. There is an expanded definition of DBA with the more robust epidemiology, with the results from the international registries. It's a rare disease, so registries are essential for taking into account the patients and following them. So we have here at the Feinstein, the DBA Registry of North America that is hosted by Jeff Lipton and Adriana Vlahos. Then you have the DBA UK, Germany, Italy, Sweden, France, and many others. Again, this is a rare disease. I'll get back to that. Gene discovery has been very helpful to expand the definition of the disorder. There are right now 23 published DBA genes, and 11 of them were discovered through our DBA registry here in North America. And what we realize now is that it's very extreme variable expressivity within and between families. It's not really inherited, unlike what we thought at first. It's familiar with autosomal dominant transmission in 45% of the cases. But all this led to the modern diagnostic criteria. It is a ribosomopathy, as I'll get to that. It's due to a mutation in the ribosome proteins, and there is a RP mutation, ribosomal protein mutation, but there are also mutations that have been discovered in GATA1 by the team of Vijay Sankaran in Boston, and other new mutations as well, such as TSR2, HETHR3, and others, which led to an expanded clinical criteria. And now we're talking more about Diamond-Blackfan anemia syndromes than about DBA. So to talk a little bit more about the clinical criteria, and what we have here in the registry is the incidence of DBA. So it's a rare disease, 7 per million live birth. You have about 25, 30 new patients per year in the United States. So again, really rare. The enrollment that we have today in 2026, I updated these numbers actually on Friday, is 898 total, with 859 alive in North America and 39 international patients that are enrolled in our registry. And the ratio male to female is really 1-to-1, 448 versus 450. So now let's dig into the molecular etiology of these syndromes. So as I mentioned, it's a ribosomopathy. The mutations are involved in ribosome biogenesis, okay? So RP ribosome proteins, S for small subunit, L for large subunit. In 25% of the cases, you have a mutation in the so-called RPS19, ribosomal protein small subunit 19. And then in others, then RPL5 is another one that is really mutated very often in these patients. When I say often, of course, is to take with precaution because it's a rare disease. But then, as I mentioned, you have non-ribosomal proteins due to faulty translation, such as TSR2 and others that are involved. But what I would like you to note here is that there are still about between 20 and 25% of the cases that are unidentified, and we still don't know the etiology of these cases. But still, even though we have therapies for DBA, some therapies, as I mentioned, the first line of treatment is steroids, okay? Then I would say that about 60% to 80% respond to steroids, but then most of the patients develop a resistance to steroid. We did a study with one of my MD Ph.D. when he was in the lab trying to understand the steroid resistance, and we know it's related to cell cycle regulation. This is something we did in collaboration with the blood center. It's involving the protein P57 KIT1. And when patients become resistant to steroids, they undergo transfusion, blood transfusion. So of course, you have to think about the side effects, what it leads to. First, the side effects of the steroids. I already mentioned the bone defects, the growth defects, but then you add to that then the transfusion. And of course, it leads to iron overload when patients undergo transfusion for a long time. But what the field is going into now is the development of small molecules. And you may have seen in the literature several papers, some of them from Lanzons, from others with the TFP and calmodulin inhibitors that are playing a role in P53 regulation. I'll mention P53 in my talk, which is why I'm already embarking on that now. And then, of course, gene therapy. But when you think about gene therapy, you have to remember that you have 23 genes that are involved, and all at least. And although there are studies that have mentioned that everything converges onto GATA1, this notion of a GATA1 short that is translated, defective translation of GATA1, there are still some questions that remain. Because as I mentioned here, all these treatments are here to solve the anemia issue in these patients. However, this is a germline mutation. And although the red cell is the tissue, the hematopoietic tissue, the erythropoiesis compartment is the one that is the most affected. It's not the only one. If you remember at first, I told you about the skeletal defect, triphalangeal thumb, and other defects where GATA1 is not expressed. So even if you go for gene therapy to correct the anemia, you are not correcting the other tissues. And so that is something to keep in mind. And I'm not going for or against any gene therapy, but it's something to think about. And so for that, what we
Neil,
need
can I ask a question?
Of course, Iqbal.
So given the fact that there is at least 23 genes
Mm-hmm.
that causes this phenotype called DBA, What is the commonality among DBA patients besides anemia?
Well,
Should
that's
be a lot of heterogeneity, right?
yeah. So that's the thing. Is there a genotype phenotype correlation? Not really. Okay. And so I'm gonna allude to that during this presentation now.
Okay.
And the problem with facing is that there is no consensus, I would say, on what is the common factor. And that's the question you and I have been talking about.
Yes. Yeah. Okay.
So I still don't have the answer from what you and I talked about 3 months ago. Although I may have one that you may not agree with, but that's a hypothesis now.
Okay. Cool. Thank you.
Okay. So as I mentioned, the big challenge that we've been facing, okay, is to model the anemia in DBA. So the human CD34 positive cells, of course, the primary cells, very few cells, shRNA, there are off-target effects. And now with Mitch Weiss and Senthal, who has now his lab at St. Jude, there has been development of CRISPR technologies to really target the gene of interest and develop some culture system. Then you have the human iPS cells. Problem with these cells, of course, is that when you go towards erythropoiesis, you go towards a more primitive lineage and differentiation. So that's kind of an issue. Then you have the zebrafish that has been mostly developed by Len's group. Here you need the complete ablation of the RP of interest. You cannot have autosomal dominant, you cannot have heterozygosity to have the phenotype. And in the mouse, you have 4 to 5 models so far. And when I say 4 to 5, it's because I'm interested mostly here in the RPS19, RPL5 in the context of what I'm going to present today. But you have to know that Jen, who's here today, has an RPL11 mouse, which has also microcytic anemia, but with reticulocytosis, which is what I mentioned here at the bottom. The mouse models, they are anemic, they are microcytic, they have an increased cell volume, but its cell volume is due to an increase of reticulocyte count, and I'll get to that in a minute. So what we did is, it was an accident. It was a total accident, actually. We have been studying RPS19, the mouse, to study the bone defect. And I will totally wipe out this for this talk because it would be too long. But during COVID, what we did is that we had the mice on the shelf. And what I said to the fellow who was in the lab at the time is, let's try to breed them with the VAV-CRE. And people were telling me, Mohan, starting with Mohan, you're crazy because nothing will happen. All the mice have stress erythropoiesis and they're gonna be reticulocytic. And I said, well, we have the mice, let's try. And so what happened is that these are the mice. The mice at P10, they were all dead. Okay. These are the mice at P6 and at P0. You see they are pale, they are smaller, they are under a VAV-CRE promoter. And so what happens is this. They have pancytopenia. I'm going to be honest. They have a white cell count that is decreased. They have the platelet count that is decreased, although it seems to recover, to have a trend to recover towards normality, towards their compared to their littermate control. However, the most striking phenotype here is that the red cell count is down with an MCV that is increased, okay? So here, for the mouse aficionado, of course, we have an MCV that is comparable to the human one because we are in the neonate mice, and I'll get back to that in the next slide. And so here the MCV is increased, so they are macrocytic, but to our surprise, they were also reticulocytopenic. And why is that? Well, here is a study that I got lucky to be involved in 2021 when I was still collaborating with Mohan, and I'm still collaborating, but that's an old story when I was in his lab. From the newborn mouse to the adult mouse, for the erythropoiesis to reach steady state, it takes about 7 to 8 weeks. For the bone marrow to be fully independent without needing the spleen or the liver. The liver is still erythropoietic for 7 days, the spleen for about 7 weeks, and the marrow takes over fully by 7 weeks. So when we look at these mice, both grossly at the hematopoietic organ, erythropoietic organ, and then microscopically, what we observed is that unlike any models we had seen so far, except for one mutant of the EPO, and I cannot recall the name of the mutant, I'm sorry about that, is that the spleen was atrophic, the liver was completely white, it wasn't erythropoietic anymore, and the bone marrow inside was totally white. And what you can observe here is the spleen, marrow, and liver. So really something totally unlike what had been observed before with other models of heterozygosity of RPS 19. So we decided to take a step back and look at fetal erythropoiesis in these animals because, and I swear I'm going to get acquainted to that. So we decided to look at fetal liver, at erythropoiesis in the liver. And for that, what we did is that instead of using CD44 versus TEL119, which in the liver is not as accurate as it is in the bone marrow, we used the method that Merab Sokolovsky had developed when she was in Harvey's lab and then had expanded and optimized for the fetal liver. So from S0 to S5, you have the erythroid progenitors, and I'll remind you that throughout the presentation. And then S2 to S5, you have terminal erythroid differentiation. And these are the mice. The mice at 13 and a half, 17 and a half. So really no big difference. Maybe, of course, the liver a little bit smaller and the total cell number decreased by 30, 40% at 13 and a half, with really a big difference, a 50% difference in the cell count for the TEL119 positive cells. And at 17 and a half, then this RPS 19 heterozygous mice, and already we know they are haploinsufficient. I don't show the Westerns here, but they are haplo. What we observed is that a 50% decrease in the total cell number, and this is due to a 50% decrease in the TEL119 positive cells. So what is happening in terms of erythropoiesis? Well, you read that clockwise, okay, for E13 and a half, E17 and a half. And what we observed, we noticed already an accumulation of the S0 S1, okay, that is even more dramatic at 17 and a half. Now, if you express them as a fold change, you have this increase, 13 and a half, but strikingly, what we did not understand at first is why we had a decrease at 17 and a half as a fold change in the expression, while at S3 we had this 50% decrease that we had already observed at 13 and a half. So here so far what we could conclude, okay, what we hypothesized and concluded is that the cells that pass this issue in the erythroid progenitors at S0, okay, they make it fine. Meaning you have 50% reduction, but then once you pass this block, you make it fine. Kind of what you observe in patients with DBA, with a mutation in RPS 19. It's less severe than a patient with an RPL5 mutation and an RPL11 mutation. The anemia is a little bit less severe. So what happened is that we tried to understand this S0 mutation. And then I called Anna that day, I remember, because I told her, I said, okay, can you tell me, because I don't understand. Hematopoiesis, okay, erythropoiesis for me is easy. Hematopoiesis is a whole mess. I don't understand. Why do you call a long term, a short term, an MPP, whatever? And she said, oh, that's easy. You do this, that, and that. I said, in the fetal liver. And she said, forget about it. Nobody really knows about that. I said, okay, so what am I going to do now? She said, well, take this and that, and it should be okay. So what we did, I followed her advice, and I did CIHD APC7 versus SCA1. And on that, took the LK versus the LSK, and from the LSK, had three populations. You can yell at me, okay, but I don't know. That's what I've been told to do. That's what the reviewers are okay with so far. So I'll get with that, and then we can nitpick all the different ones. But the long term versus short term versus the MPP population, the main message here is that indeed by 17 and a half, we had a dramatic decrease of these populations. So again, explaining why at first we had an increase of this S naught, maybe, and then at 17 and a half, a total decrease, a total depletion of it. Granted, these are not transplant experiments, but I don't know what I would transplant with this at 17 and a half. However, I'm going to show you we did some functional assays. But in the meantime, I said, why are we going to stop with an RPS 19? Let's try the RPL5 as well. Let's try to compare them. So we did the RPL5, and here what we observed is that at P1 we had absolutely no mice born. They are born maybe for 30 minutes, 1 hour, okay, but they die. They really die at birth. And so what happened is that we did the same approach. Again, I'm not very clever. When something works, I do the same for the other one. And so E13 and a half, very interesting. Same observation as the S19, 50% decrease, 50% decrease, and here 50%, 50%. Okay, so here we were happy. We said, okay, so probably the method is working and the mice are having the same, taking the same route. However, here was what we observed that was really interesting to us, is that instead of having this decrease in the S0 population, they had an increase of that. And then a decrease in the S3 population, saying that here, once they embark on terminal differentiation, they die, or they don't make it that far. And here telling us that in the S0 population, this is really where you have this issue, I would say, in the RPL5. And this, I went back talking with Mohan, with Lily Da Costa, and other people, Jeff Lipton, that are expert in DBA. And Mohan reminded me, he said, well, you know, before we had the genotypes, he said, we have the cells that grow and the cells that don't grow in culture. And granted, when we had the genotypes after from patients, the cells that grew were the ones RPS 19, because indeed, once they pass, they make it. And the ones that don't grow were the RPL 5, because they die. And at the S, it was not S, not S1. And it's even more complicated in humans. So we were feeling good because we were saying, well, maybe this validates our models. And so we look the same way at this long-term, short-term MPP LSK population. And what we observe immunophenotypically is an increase of this compartment. Is it functional? No. When we transplant them, okay, they die. But to compare with the RPS 19, what we really did is the colony forming assays. And what you can observe here is what I told you before, basically, is that the BFUE, and here, very interestingly for us, and that reflects on the blood counts as well, is that they are not making any colonies. The LSK compartment, the stem and progenitor cell compartment is really affected. And in the case of the BFUE, compared to the littermate control, and now we're confronting them and doing some more elaborated stats, but I can tell you that in the case of the RPL5, it's more dramatic here. You can observe it here actually compared to the RPS19 for BFUEs than it is for the RPL5. Okay, so now we have a model that seems to replicate clinical findings in DBA in patients for both RPS19 and RPL5. Can we address partly at least Iqbal's question and others, okay, what is happening in these guys? So what we did is that we looked at translation. Okay, we did some polysome profiling and some OP-Puro assay. I talked to Rob Signer about all this and to look into this because again I wasn't really expert on that, and to an old friend of mine as well who's in France, and Sebastian, who has been helping me on global analysis. So here, the same, it's even more complex. So we took the c-KIT positive population versus the TEL119 positive, because when you think that you need about 8 million cells, okay, to do these experiments, forget about it. Impossible to do any experiment on the sorted populations or on the so-called long-term, short-term MPP. So let's keep it easy and take c-KIT and TEL119. That unravels already some data and some interesting results. So for those of you who are not really aware of this method, the polysome profile is basically just a gradient, okay, where you load your lysate on top, and then you let them sediment with the help of an ultracentrifuge. And then you look at the 40S, S for Svedberg, the sedimentation unit. And 40S is the small subunit, 60S the large, 80 is the monosome, and then you have your polysomes. Long story short, what we observed is that in the c-KIT, we had the defect, okay, of RPS19 in blue, L5 in purple. You'll have this all along. We observed that in the c-KIT and in the TEL119, we had the defects that we were expecting, meaning that in RPL5, the defect was really maintained all along. But in the OP-Puro, we had surprisingly an increase here in the LSK population. So this we could do it, the OP-Puro, because it's done by flow cytometry. We could gate for the LSK population. And so we had an increase in fluorescence, meaning an increase in translation. So this was surprising to us because, of course, you have an issue in translation. Okay, here you see in blue, in purple, the decrease in the polysome, meaning activity of translation. So why do we have this increase here? So then you go in this world. You go in the world of elongation, initiation, cap-mediated translation. We translate, I think, about 80% of your RNA versus the IRES and the stress translation. It's a whole world, okay? And here, long story short, you can look deeply, and I spared you the whole Western blots. But here, what we observed is that the cap-mediated translation factors, okay, were whether increased, okay, you can observe here, this one is not statistically significant, or unaffected in RPS19, while they were decreased in RPL5. Look at E, for example, here, EIF4G, this is 17.5, they were all decreased. And then there was a transient activation of the stress response in RPL5, which could explain maybe why you had this increase at first that we noticed in the mice. And then Naomi Taylor published a couple of years ago in Blood a very interesting study showing the hyposynation of EIF5 alpha in the metabolism and in erythropoiesis And so we looked at hypocyanation. And what we observed very interestingly is that hypocyanation appears to be model specific, with a role mostly in RPL5 more than in RPS19. And we are currently investigating this. So, having said that, what we did is we decided to take a more global approach. I said, enough. We're not going to investigate all these factors one after the other. Let's go global. And so we did a polysome sequencing because at the time we didn't have enough material to do ribosome sequencing. And so polysome sequencing was helpful in that case. And so here it's very easy. If you go through the slope like that, if you follow the slope, you have equivalence, okay, between what is transcribed, cytosolic, versus what is translated, poly. And what we observed is that in the TEL119 compartment and in the c-KIT compartment for the LPS19, okay, there were many differences. And what you can observe here for the RPS19, RPL5, and here the c-KIT is that there was, here I'm going to take this one or even this one, there was a transcription element that was apparent. And this to us was something new because, of course, we were saying, okay, this is a ribosomal protein, it's involved in translation. Why would there be a transcription defect as well? Well, what we think is that it's secondary, okay, to that. But nevertheless, it led us to do some single cell RNA-seq on these guys, on these mice. And so single-cell RNA-seq revealed what? Well, the cluster distribution first between RPS19 and RPL5 was very cool because first, the HSPC compartment in terms of gene signature, in terms of cluster, so of course not phenotypically or functionally like we observed before, was increased. Okay, well, it was not really affected in the RPL5. However, the erythroid cluster was the most affected. But this really is just to, I would say, to illustrate a figure in the paper. Now, I haven't seen a paper without this kind of figures in some time. So what was really cool is this. We did a pathway analysis comparing RPS19 and RPL5, and I have to give all the credit to John's team on that because they thought about doing this. And what they found is actually something that reconcile many different hypotheses here. Is that what Jen had been publishing with the heme metabolism, okay, with her models and with the heme imbalance and what happens, well, it was highlighted here in the proerythroblast. The GATA1 targets are present. You name it, we could find these different pathways. But then at the individual gene levels, there were some surprises. For example, if you look at genes that are involved in apoptosis, like P21, like BAX, we only found them upregulated in the RPS19, not in the RPL5. So what does it mean? So we decided to look after that and try and understand what is happening here. So, of course, the first step is to look at the cell cycle, right? Because if you think about P21, about BAX, you think about P53, and of course, this is the paradigm in Diamond-Blackfan anemia. In ribosomopathy, you have activation of P53 due to cell cycle, due to the G1 arrest and the cell cycle stress. So we undertook that, and we did some measurements. We followed Merav Sokolovsky staining protocol with EDU and DAPI to measure G1 as G2M. What we observe is indeed we have a G1 accumulation, okay, in the LSK. not in the 10-119. So again, reminiscent of what we had observed phenotypically, and once they pass this step, they are fine, they're okay. So it made me a little bit more comfortable. While in the RPL5, we had this G1 increase in the 10-119. So again, this dichotomy, and in the phenotype, everything an effect more on the LSK RPS19, hematopoietic defect, while pure erythroid defect in the RPL5. And then what we did is that we took a measure of the MFI for the S phase. So I remind you, Merav published a very elegant study showing that when the MFI increases, okay, they cycle more rapidly. When they decrease, they cycle more slowly. And here we found total opposite phenotype when in the 10-119 or in the LSK, the opposite, they cycle faster in RPS19, they cycle slower in the RPL5. So what does it mean in terms of P53 and what I told you? Well, by bulk RNA-seq, what we observe is an increase, okay, in RPS19. So something that we wouldn't expect. So maybe this is an anomaly or something in the mouse, because P53 is usually increased at the protein level, not at the RNA level. But really, here is the message, is that in the sick population, we had a dramatic increase, okay, at the protein level in P53 for the RPS19, while we had comparable levels of P53 in the TEL119 positive population. So what does this lead to? Well, here was the big surprise at the time, and this now has been comforted somehow by a publication from Vijay and his group showing that there is ferroptosis in different models of ribosomalopathies, is that in the RPS19, there is apoptosis of the progenitors, okay? However, in the RPL5, there is absolutely no apoptosis. It's actually decreased, and we have other markers, of course. However, when we started looking at the markers for ferroptosis, we observed an increase, both in the c-KIT and in the TEL119, even more. So how do we validate this? Of course, we can do microscopy, we can do some elegant staining. I mean, some of you here and Brita, others have done it. But we had already the single cell. So we went back to the single cell instead of pulling more reagents. And here are some, on the first line, some pro-ferroptotic, and at the bottom, some anti-ferroptotic, blue RPS19, purple RPL5. And what we observed is that here is the best example, the anti-ferroptotic were up, while we had no change or decrease in the anti-ferroptotic in the RPL5. This was totally validated by Western blot. Here is an example here, okay, and here for the PCBP1 over there. Okay, so now we've validated that RPS19, they go for apoptosis, RPL5, they go apparently for ferroptosis. So how can we understand the role of P53 in this whole thing? So of course, what we did is that we remove P53 in the system. So we remove P53, whether one copy or two copies in the RPS19 first. And so what happens? Here it is. This is the double heterozygous, okay? So you have one copy of RPS19 removed, one copy of P53. The mice are still microcytic, the blood count. However, they become they have more reticulocytes. So to me, it means that probably the spleen indeed is becoming erythropoietic. And it is. You have an increase in terminal differentiation. However, for some reasons that we still don't know why, and this is something that we're going to investigate in the future, and I'm happy to hear any feedback, any suggestion, the bone marrow is still empty. There is no dose-dependent effect of removing one allele of p53 in this model. Only stress erythropoiesis is rescued. Now, if you remove two copies, so this, the green, you remember that's what I was telling you, in the fetal system, if you remove two copies here in red, you rescue everything to normal. And then the bone marrow, everything is back to normal. So what is happening so you rescue the numbers. What is happening in the RPL5? In the RPL5, the same. You remove one copy of P53, you don't rescue anything. They don't even reach birth. So meaning that, I don't know, they don't even get to the stress erythropoiesis point. So this is something that, that I need to understand and I need to think about more. But then you remove the two copies, you're fine. Here it is in orange. Totally fine. You rescue the capacity to differentiate in the late stages. Okay. So one last thing in the system, in the model. What can be happening in these mice? Is P53 governing everything? What is it? So here I was looking at our single-cell RNA-seq and our polysome sequencing and doing more analysis. What we observed is this thing that was peculiar at first, is that in terminal differentiation, we had increased expression of the transcription factor RUNX1, which is totally unexpected, okay? Because RUNX1, as you know, once you pass the MEP stage, goes down in red cells, goes up in MEGs, okay? So then I went back to the literature. And of course, I found Rob's paper, Nancy Speck's papers that said that it plays a role in ribosome biogenesis. And so I said, okay, so maybe there is something here. Again, total hypothesis, I don't know. So I went back and I looked at the polysome sequencing, and indeed, in the c-kit of the RPS19, so this is the reactome. This is not the gene itself or the protein itself. So RUNX1 regulates transcription involved in differentiation of hematopoietic stem and progenitor pathway. Here, while it was not changed, cyto is the transcription, okay? The pathway wasn't changed. It was changed at the translation level, sorry. And so then we look by Western and look at this in the c-kit population compared to the controls. While in the RPL5, there was no change at all. So what does it mean? We did the same. We removed RUNX1. We used the RUNX1 flux. And here's something very interesting, I would say, but that doesn't bring a lot more to the table, is that in using the same gating strategy and the same markers, we observed an increase by removing one copy of LOX1, but this was not dose dependent. It was maximized with one copy of RUNX1. We observed a correction, okay, of the quantity of MPP and stem progenitor. However, terminal erythropoiesis was even worse, which is something that has been described in the literature a long time ago. So I don't really know how to exploit that. We have the colony forming assay. We did them. Indeed, there is a little bit of rescue, but it's not dramatic. And so I think it plays a role, but I don't know where at this stage, except that we had done some multiomics and some single-cell ATAC-seq. And then we go back to GATA1 and what I was telling in the very beginning with the gene therapy and other things. Well, we have different transcription factor networks that are involved, okay? Because when you take the single-cell ATAC for the RPS19 logs, or the lux plus one copy of p53 in that case. What you observe by analyzing with Systrom and all these databases is that p53 is, of course, the first one, but you have all these transcription factor pathways, regulatory pathways that are involved, upregulated or downregulated. And yes, GATA1 is here, upregulated and downregulated, but RUNX1 is here as well. So this is going to be published soon. You're free to take a screenshot if you have your favorite pathway to look at. But we looked at RUNX1 in this context. And indeed, RUNX1 can bind to P21, going back to what I was telling you before. And indeed, when we remove RUNX1, okay, here you remove RUNX1 completely, P21 goes down, but P53 remains up. However, when we remove P53, in the context of haploinsufficiency still, okay, should have mentioned that, you remove both P21 and RUNX1. So what we think now is that RUNX1 is acting between P53 and P21. But of course, there is a lot that remains to be done. What is the relevance to humans? My friend Dina Iskander in London had published this Science Translational Medicine showing the single cell in patients with DBA. And I told her, I said, did you come across RUNX1? And she told me, yeah, but we didn't know what to do with it. She said, why? I said, well, because now we see it in the mouse and it seems to be very specific and acting between P53 and P21. Can you go back to your data? And she did. And she looked at them. And indeed, RUNX1 is increased in DBA, in the HSPC compartment and in the erythroid compartment only. And when you look at patients, this is a capillary western, okay, this is called the JES, I'm sure you've heard of it. It's indeed increased in patients with small subunits, mutations. So when you think about RUNX1, when you think about cancer predisposition, you're thinking now, well, maybe we could have another link here, but this is speculation at this stage. In summary, before I take it for cancer for the last 2 minutes, what we have is what we think is that from the HSPC to the EP to the red cell is you expand your pool of progenitors and your pool of red cells. While in haploinsufficiency for RPS19, we think that the defect is really at the HSPC, okay, due to activation P53, RUNX1, apoptosis, while in the RPL5, it's a defect at the erythroid progenitor due to ferroptosis, which could also explain this heme imbalance, these issues that we observed in other models. That's to be exploited. But now, if we go back to cancer, you look at cancer in DBA, and the gene mutated with patients, what I showed you at first, is different ribosomal protein, including RPS26. If now you superimpose it to patient with cancer and or MDS, what you noticed is that RPS26 is missing. None of the patients have had a mutation in RPS26. So what we've been doing is that we looked in the hematopoietic model, and of course, what we've been doing is that we've removed P53 as well. And here, this is a 8-month-old mouse representative. We have more mice, of course. This is what happens. And you observe beautiful cancer in the peripheral blood of these mice, while the P53 do not still. This one, this is just blood smear, okay? So we let them age, and they develop cancer, these guys, around 10 months, while these ones did that already. Then you look in the bone model that I didn't present you with, and they have osteosarcoma. So of course, then you're thinking, what happens to an RPS26 model? So, oops. we created the RPS26 model. And while I don't have the cancer part yet, I left that for because it's an iron seminar as well, for people to look into this. We still have an increased MCV compared to the RPS19, a decreased retic count. However, the MCHC is down. The MCH is down, hypochromic and microcytic. And that we're going to look at it. The spleen is smaller, but we don't know why. Okay, so with that, I'm going to conclude and thank everybody who did the work. Yufeng has been taking the whole project with the help also of Alexi and John's lab and other people involved and the people that contributed to the work. Julian has been working with me for more than 15 years now, our funding sources in the lab and patients and family. And thank you for your attention. I'm happy to take any question now.
Thank you, Leo. Any questions? You can just unmute yourself and
Leo, I got a mechanistic question.
Go for it.
So, given the fact that these ribosomal subunits, I'm going to assume are part of the same large subunit complex, why do you see a different phenotype between 5 and 19 at the complete molecular mechanistic level? There is no debate about the phenotypic differences. Are they recruited for different genes differentially? Because you're not making a point mutation, right? You're essentially deleting one allele, right? So haploinsufficiency is haploinsufficiency regardless of whether it's 5 or 19. So does that mean that some genes require more amounts of 19 versus 5 or vice versa? I'm trying to understand what's happening in the cell.
So here you're raising the $1 million or $2 million question. That is this a question of specialized ribosomes or the concentration model? I am in favor of the specialized ribosomes, but again, I don't know. So what we're doing to address this question now is really by comparing these models, we're doing some ribo-ITP, which is the model, the ribosome sequencing. We're looking at 100 cells. You can take now and that's what Alexi is doing, trying to address these questions. But really, we don't know. So you really have two schools here. Okay, and nobody has ever showed one way or the other.
In your polysomal profiling, right, old-fashioned, right, do you see any differences or can you measure the number of subunits within a complex and whether they change over time as the cells differentiate from HSCs to erythroid precursors?
Oof. Wow. Come do the experiment.
Mohan is laughing.
Although I must say one thing, if you don't mind.
Go ahead.
Lionel and I have been working with colleagues in Institute Cochin Proteomics. What we found quite surprising, we had validated and all, is the copy numbers of the various ribosomal subunits are very different, going from CD34 all the way to polychromatic. So I don't think I would have expected similar numbers throughout the process, but we are huge. And the pattern fits. There's a difference between RPL 19 and RPL 5 in terms of the ribosomal copy numbers in absolute quantification. Yeah, we have no idea what the hell it means, but at least it seems to be not universally same number of ribosomal proteins at all stages of differentiation. Yeah.
Yeah, you're talking about copy numbers in the cell, right? Not
Yeah,
the
absolutely.
sample
No,
that
in the
polysome.
purified populations of erythroid at 9 different stages of differentiation. Yeah.
But not in the polysome, yeah.
That was going to be my question. So the ribosomal components are not expressed similarly in all the different populations.
Especially during erythroid differentiation. That's, yeah, absolutely.
I have to say though, I'm just as confused as I was 3 months ago, Leo.
Hey, that's science, man. That's science. Data is data.
Yeah, so that's what I was going to go along with. Data is great, and thanks, Leo. Those are great. And as you probably know, we've done a lot of single cell work in DBA patients, and also with models that where RPS 19 is knocked down, and the mouse only RPL 11. And so I've never dealt with RPL 5, and I'll just put that in a context. And so it was rewarding to see very similar data. And what I should say in patients, just to make things a little more confusing, is we've always saw a halt of differentiation in a single cell model, and whether it was attributable to apoptosis or ferroptosis, we saw both, and we never tried to sort it by genotype. And so I find that actually intriguing, that the mechanism, we always just call that they die by apoptosis and ferroptosis, or ferroptosis, something broad like that, and never have tried to dissect it. And so I think that they both are going to do with iron and heme, and Iqbal will be happy about, and ROS. And I think it is going to be dependent on the actual on the different parameters you're looking at. And in each model or each patient, it's going to be a little bit different and lead to the same broad physiologic phenotype, but the exact mechanism and exact stage is going to depend on the amount of ribosomes and present the different kinds of defects. And so I think you've linked on to a really nice thing about heterogeneity and some of the phenotypes that have to do with just the variables you're looking at. So thanks. I was actually going to ask a question very differently, though, from another half of my brain, which is interested in cancer development. And that is that cancers broadly, and particularly solid, as well as solid tumors, as well as leukemias, are from, as you're probably aware, from P53 mutation or P53 decrease, or from RUNX1 mutation, RUNX1 decrease. Great literatures on that. I think you have a really interesting model to figure out why. Not just to knock it out, but to look at some of the mutations and their outcome, because I suspect you'll find them specific to different, begin to answer maybe some of the questions about why there's osteosarcoma or why there's a different cancer outcome. I'm not exactly how to connect the dots for you, but I suspect the literature would give a fair bit of guidance about what might connect to what. And what you're essentially looking at, as I think about what you're saying, is those two molecules are protecting cells from, they're stopping differentiation, so they're protecting them from rampant replication. And if you want to just look at it very simplistically, and there'll be very specific pathways where that's released, leading to specific outcomes in terms of the cancer predisposition. And I suspect you could find neat data either in patients or in mice about that.
Well, we have the biorep, right? That's why
you have the biobank. I mean, you have the biobank, you have the data set. I think you have cool questions.
Yeah. Thanks, Jen.
Any other questions for Leo? No? All right. Thank you so much, Leo. That was really a beautiful talk.
Well, thank you.
Thank you for joining us.
Thanks.
Nice to see
Thanks.
you.
Have a good one.
So much.
Bye, everyone.
Bye.